Imagine I tell you that one pathway of the glucose metabolism has three completely different jobs. First, it helps in our body to detoxify drugs and bilirubin. Second one is in many animals it produces the vitamin C, and the third one it forms pentose sugars that enter the another important metabolic pathway, that is the HMP. Now here is the interesting part: humans can do the first and third jobs perfectly, but we completely fail to making the vitamin C. Why? That's exactly what you will understand in today's lecture, and once you understand this pathway you will never confuse it in your university exams again. Before learning the pathway, let's understand why examiners love asking it. The pathway is important because it connects carbohydrate metabolism, detoxification, vitamin C synthesis, and the pentose metabolism. Questions commonly asked include the what is uronic acid pathway, why can't humans synthesize vitamin C, draw the pathway and explain the essential pentosuria and clinical importance of the glucuronic acid. So don't just memorize the reactions today, understand the logic behind them. First question: what exactly is the uronic acid pathway? Think of the glucose like a student after MBBS. One student becomes the physician and another becomes surgeon. Another becomes researcher. Similarly, glucose also has different career options inside our body. Most glucose enter glycolysis, some enters glycogenesis, some enters pentose phosphate pathway, and some small amount enters in the uronic acid pathway. So this pathway is simply alternative oxidative pathway of the glucose metabolism. It is also called as glucuronic acid pathway because one of its most important products is glucuronic acid. But why does body need glucuronic acid? This is where the uh, this is where students usually make a mistake. They think glucuronic acid is just another intermediate. No, it has very important function that it helps to conjugate the bilirubin, conjugate the substances so they become water soluble and can be excreted. That examples are there: bilirubin, steroid hormones, mini drugs, and toxic compounds. So whenever the liver wants to eliminate the something from the body, glucuronic acid becomes extremely useful. Whenever you hear the word glucuronidation, immediately think the detoxification. Let's discuss about the site of the pathway. Now where all these pathway occurs? Here, to small extent, it occurs in adipose tissue and mainly in the liver. And remember, all these reaction occurs in the cytoplasm of the cell. Here is the liver, it is the major site, and adipose tissue is minor site, and cytoplasm is the cellular location. Starting product is a substrate is the glucose 6-phosphate. Every pathway needs the starting molecule. Here it starts with glucose 6-phosphate, and so the same glucose 6-phosphate that can enter the glycolysis or pentose phosphate pathway can enter in the uronic acid pathway, one molecule and different destinies. End product. Now here's the interesting question: what does the pathway finally produce? Actually, there are the two important end products depending on the organism. In humans and most mammals, xylulose 5-phosphate is formed. This molecule can enter in the HMP pathway, linking the uronic acid pathway with the pentose metabolism. But in the lower animals, the additional product is formed, that is the vitamin C. Animals like goat and other mammals can synthesize vitamin C on their own, so they don't need vitamin C in their diet. Vitamin C becomes essential vitamins in the humans, so we must obtain it from the food. Here is one memory trick: GGP, guinea pigs, great apes, and our we people lacks the enzyme L-gulonolactone oxidase. So we should all require the dietary vitamin C. Let's discuss the pathway. Now let's begin the actual reactions. Don't try to memorize them immediately, understand the story. Step 1, we start with the glucose 6-phosphate, and first glucose 6-phosphate is converted into glucose 1-phosphate by the enzyme phosphoglucomutase. It is something you have already studied this reaction in glycogenesis, so nothing new here. And next, Glucose 1 phosphate reacts with UTP and forms UDP-glucose, the enzyme responsible UDP-glucose pyrophosphorylase. Students often wonder, why attach UDP? Think of UDP like delivery tag on the courier parcel. Once attached, glucose becomes activated molecule, and then it can participate in many biochemical reactions. Activated molecules are much easier for enzymes to use. Now comes the key reaction. UDP-glucose is oxidized by the enzyme UDP-glucose dehydrogenase, and two molecules of NAD are reduced to NADH during the oxidation, and the product is formed UDP-glucuronate. This is the activated form of the glucuronic acid, and this activated form is what the body actually uses in conjugation reactions. Remember the enzyme carefully. UDP-glucose, uh, UDP-glucose dehydrogenase. This frequently asked in Viva questions. Here, in the next step, second step, conversion of UDP-glucuronate to the L-gluconate. The first thing that happens in that UDP-glucuronate loses its UDP portion, and then this occurs by the hydrolytic reaction. Once the UDP is removed, the product formed is D-glucuronate. And now D-glucuronate doesn't stay like this for long. It reduced by the NADPH dependent reaction to form L-gluconate. Notice the reducing agent here is NADPH, not NADP. Many students confuse these two. A simple way to remember is NADPH is the mainly used in biosynthetic and reductive reactions, and this one of those reactions. Why is L-gluconate is important? Now you may ask, why is L-gluconate such an important intermediate? Because it acts like a crossroad. From here, the pathway has two possible fates. One can lead to synthesis vitamin C, and others leads to xylose formation. So whenever you see the L-gluconate, immediately thinks this is the branching point of the uronic acid pathway. Let's follow the first branch. L-gluconate undergoes the cyclization to form the L-gluconate lactone. And here, next comes the most important enzymes of the entire pathway, that is L-gluconate gulanolactone oxidase. This enzyme converts L-gulanolactone into the 2-keto L-gulanolactone, and in the presence of the oxygen, and finally 2-keto L-gulanolactone is converted into ascorbic acid, makes the vitamin C. And now you know how most mammals make vitamin C naturally. But here's the twist, humans cannot complete these pathways. Why? Because we lack the enzyme L-gulanolactone oxidase. Without this enzyme, the pathway stops L-gulanolactone. No enzyme means no reaction means no vitamin C. That's why humans and other primates and guinea pigs must obtain vitamin C through the diet. Imagine one patient who eats very little fresh foods, vegetables for months. Gradually, vitamin C stores become depleted. What happens? Collagen synthesis becomes defective, blood vessels become fragile, patient develops bleeding gums, poor wound healing, and loose teeth. This disease is called as scurvy. And remember, scurvy is not because of glucose metabolism is abnormal. It happens because human cannot synthesize vitamin C and dietary intake become insufficient. So let's discuss about the fourth step, that is the L-gulanolactone is converted into 3-keto-gulanolactone and then decarboxylated to pentose, and here is occurs in our human body. Here this is the necessary step, because the D-xylose, after getting phosphorylated, it can enter the hexose monophosphate shunt in the pentose phosphate sugars metabolism pathways. This is the whole reaction, that is the glucose 6 phosphate converted into glucose 1 phosphate by the phosphoglucomutase. Then reaction with UTP forms UDP, and UDP-glucose by UDP-glucose phosphorylase, and UDP-glucose converted into glucuronate, UDP-glucuronate. and then converted into D-glucuronate by the glucuronide, glucuronidase, and then reduction occurs there, and L-gulanolactone is formed. Now stop there, students. This is the branching point, and from here, from right side, L-gulanolactone is then converted into L-gulanolactone oxidase by the L-gulanolactone oxidase, 2-keto L-gulanolactone, and finally forms the ascorbic acid vitamin C. This branch is absent in humans because the oxidase enzyme is missing. Here, next part occurs in the humans, xylitol, then D-xylose and xylulose 5-phosphate enters in HM patient. Here is the next fate for the L-gulanolactone. Humans cannot complete that branch because we lack L-gulanolactone oxidase. Now is the question, what happens to L-gulanolactone in the humans? Here, L-gulanolactone is converted into 3-keto L-gulanate, and L-xylulose is formed and it converted into xylitol and is converted into D-xylulose and finally xylulose 5-phosphate. This is why the uronic acid pathway is linked to pentose metabolism. Now ask yourself, can L-xylulose cannot directly enter HM patient? No, HM patient uses D-xylulose 5-phosphate, not a L-xylulose. So first the body has to convert the L form into D form. This happens in two ways. That is converted into xylitol with the enzyme xylitol dehydrogenase, and then it converted into D-xylulose finally. The uronic acid pathway is not the isolated pathway. It communicates between other metabolic pathways by producing L-xylulose 5-phosphate. It provides another route through which glucose contributes to the pentose sugar pathways and supporting nucleotide synthesis and other metabolic processes. Next, we will discuss about the effects of drug on the uronic acid pathway. Now, let's move on the important clinical application. Many drugs are metabolized in the liver by glucuronidation. When drugs such as barbiturates and chlorobutanol are administered repeatedly, the liver increases the activity of the uronic acid pathway. Why? Because the body needs more glucuronic acid to conjugate and eliminate these drugs. So, drug administration can increase the glucuronate synthesis from the glucose. Interestingly, some drugs like aminopyrin and antipyrin, these are these drugs when have these interestingly some drugs like aminopyrin and antipyrin have also been shown to increase the vitamin C synthesis in animals. Remember the word animals because humans cannot synthesize the vitamin C. And here is the tip that if asked why the certain drugs stimulates the pathway, answer is to increase glucuronidation for the detoxification. Here is now let's discuss about the one of the favorite viva and university exam question, that is the imagine a patient whose urine contains a large amount of L-xylulose. What could be the reason? The reason is the essential pentosuria. What causes it? It is the rare inherited genetic disorder and defect in enzyme of the xylitol dehydrogenase, which is the NADP dependent enzyme. Normally, L-xylulose is converted into xylitol, but if the enzyme is deficient, L-xylulose cannot be metabolized further, and as a result, it accumulates and excreted in the urine. And here the clinical features that here interesting part is the most students think that if metabolic pathway is defective, disease must be severe. Not here. Essential pentosuria is usually asymptomatic and individuals suffers from no ill effects. The main finding is the presence of L-xylulose in the urine, and so don't confuse it with diabetes mellitus. In the diabetes, glucose appears in urine. Here, L-xylulose appears in urine. And your notes also mention an important point: administration of aminopyrin and antipyrin increases the excretion of L-xylulose in patients with essential pentosuria. This is an important exam fact. Here we have covered the all uronic acid pathway. Here is one MCQ for you. A patient is unable to synthesize vitamin C due to absence of which enzyme? Answer is the L-gulanolactone oxidase. Today you didn't just memorize the uronic acid pathway, you understood why it exists, how it connects detoxification, pentose metabolism and vitamin C synthesis, and why humans are different from most animals. If this lecture helped you understand this concept, subscribe and because we are completing other topics also. Thank you.